Good morning, good afternoon, and good night. Welcome to the lecture on interim analysis, adaptations, and master protocols in the intermediate courses of the EGP. My name is Franz König. I'm a biostatistician at the Medical University of Vienna. Here in Vienna, I'm also a member of the ethics committee, and I'm head of a working group on adaptive designs at the Center for Medical Data Science. Here you can see what I will cover today. So first, I will give a very quick introduction. And in today's talk, we will really focus on interim analysis, on group sequential designs, how to do adaptations, and in the very end, you will learn something about master protocols. And today I will focus on confirmatory clinical trial, which are mainly based on frequentist tools. I will explain it later on. So today I won't cover Bayesian designs, as you had already a lecture on Bayesian designs itself. So how do most classical drug development programs look like? Usually we conduct several clinical trials, starting from phase 1 trials up to phase 4 trials. Conventionally, at least 2 phase 3 trials are conducted. The phase 3 trials we also call pivotal, the confirmatory clinical trials. However, in rare diseases, we are sometimes happy if we have one trial at all. There might be sometimes some studies in parallel, either from the same drug or from different drugs, but each trial stands on its own feet. Each trial with its unique design, and each has, for example, its own control, and there's traditionally no sharing of control data across trial. So here you could argue that traditionally we learn a lot about controls. And as said, today we would like to focus on confirmatory clinical trials. So phase 3 studies are called the confirmatory trials. And here you really want to have a proof that your drug is efficacious. And regulators really care about these confirmatory studies, as they don't want to approve a drug which doesn't work. So they really care about that the false positive rate is kept small. This means that the vulnerable rate should be small and be controlled at the pre-agreed level alpha. Usually we use 5% when we use a two-sided test, or an alpha of 2.5 when we use a one-sided statistical test. And traditionally, the multiple step vulnerability is controlled at study level, regardless how many and which other studies are performed. However, you can see as there are some trials in parallel, whenever new data arises, we want to learn from a new data when they become available, either from outside the trial or inside the trial. So this means if the trial is still ongoing, there's a demand for interim analysis and adaptations. However, the traditional clinical trial paradigm says plan your clinical trial, then behave like the 3 little monkeys where you have to close your eyes, your ears, and your mouth. You're not allowed to look at the data. Stick to the initially fixed study design, and only at the very end you are allowed to analyze your data, but you are not allowed to learn in between. And as we are speaking mainly about adaptive design today, let's first have a look how traditionally the decision making is done in such a confirmatory clinical trial. As said, today we will focus on frequentist methods. So traditionally, frequentist hypothesis tests for decision making are used in confirmatory clinical trials. So the null hypothesis might be stated in a way like the experimental treatment is not superior to control, which is tested with a statistical test. And based on the clinical trial data, a p-value is calculated at the very end. And you have seen it before, whenever the p is smaller, the p-value p is smaller than 0.05, the null hypothesis can be rejected and the drug is declared as efficacious. And by doing so, this guarantees that the probability of a false positive result, given the treatment does not work, is controlled by 5%. However, if multiple tests are performed with the same threshold of 0.05, the risk of at least one false positive conclusion increases. And for example, if you have time, you might go to some of the statistical guidelines which are available at the EMA or FDA. And for example, if you look at the points to consider document on multiplicity issues issued by the EMA, it says control of the study-wise rate of false positive conclusion at an acceptable level alpha is an important principle and is often of great value in the assessment of the results of confirmatory clinical trials. So, as said, we would like to learn from the data. That's the reason why we are interested in doing an interim analysis. And the right upper corner, you can see the trial. So I will start with a 2-arm trial, and you can see halfway through, we would like to make an interim analysis. So what's actually an interim analysis? An interim analysis is any analysis, summary, or inspection of unblind trial data during an ongoing trial. Why are we interested? So first, for ethical reasons, because we would like to stop the trial if there are any ethical concerns. But also, we would like to stop the trial if you have an overwhelming effect so that patients outside the trial can benefit from the treatment as well. But not only ethical reasons, there are also economical reasons. If I can make my decision earlier than pre-planned, clearly I can save resources and can use these resources to conduct additional or other trials. So we would like to foresee the possibility to stop the trial early on the one hand side for safety issues, or if we see an overwhelming effect, or the other way around, we would also have the possibility to stop the trial for futility, meaning there's a lack of efficacy. For example, if my new drug is worse than placebo, we would like to stop for futility because I don't expect that for the remainder of the trial, the negative effect is overturned. And as we look into the data anyhow, then it's very tempting to learn from the data itself and redesign your trial. For example, in the beginning, when you write your study protocol, you have to provide a sample size calculation, and you all know how hard it is to do a sample size calculation because you have to take some assumptions on the expected treatment effect or on the variability of the data. And for example, if you see that the variability is slightly larger than anticipated, then you know your study would be underpowered, and therefore it would be a good strategy to increase the sample size accordingly so that overall you achieve the targeted power. Good. So now we're in the situation where we would like to do an interim analysis. And the first question what comes to our mind is, what will happen if we ignore the pre-planned fixed sample design and use the conventional level alpha test to decide whether to stop or continue a trial? So this is a magic p smaller 0.05. Clearly, if I do the analysis at the very end, so here we have a trial, for example, with a pre-planned sample size of 50. If we do one analysis at the very end, we use a p-value smaller than 0.05, everything is fine. Overall, the type 1 error is controlled. Now you can see if we add one interim analysis and also in the interim analysis, we use the decision rule p smaller 0.05, then instead of controlling the type 1 error rate at 5%, out of a sudden, the overall risk is already increased up to 8%, which will make regulators quite nervous. And now you see, the more interim analysis we conduct, the higher the type 1 inflation actually becomes. So for example, if we do 3 analyses each at full level alpha, we are already up to 10%. And clearly, the more interim analysis we do, the higher the type 1 error inflation becomes. So when we talk about confirmatory clinical trials in RDC, despite any design adaptations, we are still keen on controlling the type 1 error rate. Not necessarily at 2.5%, it could be higher as we are in the rare disease setting. So what are the main principles? The first methodology, which is now commonly used in clinical trials, are group sequential design, which means here you design your trial with multiple interim analyses over the course of the trial, and we predefine a set of critical boundaries to account for multiplicity. And the overall boundary crossing probability is maintained at a pre-specified significance level alpha. So which means instead of using this 0.05 in each interim analysis, we decrease the local significance level as long as so to reach overall the targeted level alpha. So for example, in the 70s, typical group sequential design was suggested, one by Pocock, the other one by O'Brien and Fleming. And on the right-hand side, you can now see how they work. Like when you would foresee for 4 interim analyses and the final analysis, instead of using p smaller 0.05, then you would have to use p 0.0158 if you use a Pocock design. So a Pocock design has the characteristic that we have the same local significance level at each interim analysis and the final analysis. However, quite quickly, people realized that these designs are not the most efficient ones, because it means you spend the same amount of the significance level regardless how many observations you have already collected. And therefore, the O'Brien-Fleming designs became quite popular because they follow the philosophy that the more data you have collected, the more alpha can be used. And you can see here, again, when you do 4 interim analysis and the final analysis, in the very beginning, you have quite a small level alpha. So the p-value has to be smaller than 0.001. And later on, the significance level becomes higher and higher. And you can see in the final analysis, the p has to be smaller than 0.0413. So it's already quite close to the conventional 5% level if you reach the final analysis. And the good news of O'Brien and Fleming designs in this respect is, as the final significance level, the adjusted significance level, is already quite close to the targeted level alpha, it means not much has to be invested in terms of sample size. So the maximum sample size of an O'Brien and Fleming design is quite close to a fixed sample design compared to a Pocock design, which would require a quite substantial increase of the maximum sample size to reach the same power. And these days, if people conduct a group sequential design, they are mainly using alpha spending function with O'Brien-Fleming type of designs, because the original group sequential designs by Pocock and O'Brien-Fleming required equally sized stages, but with alpha spending function, as I will explain in a minute, it doesn't matter when you perform the interim analysis, and also the interim analysis can be with different sample sizes. So this is the first class of designs, and adaptive designs build on group sequential design, but in addition, they also allow for design modification during the trial. So you can change the randomization allocation, you might change sample size, you might perform a treatment selection based on the interim results, but you can also utilize any external data available. For adaptive designs, the study protocol becomes more complex because you have to describe the decision rules and the criteria in the protocol so that the changes are really made by design and not ad hoc. And for the analysis, you really have to pay attention to use specific statistical methods to address the adaptations. And later on, I will explain the concept of adaptive combination test. And if you're interested to calculate such boundaries yourself, there are a lot of software packages out there, East, AdPlan, and Query, where you can calculate these boundaries. And there are also freely available software out, mainly in R. You might have a look at this Shiny app from RPact, where you can easily calculate such boundaries in dependency of the number of interim analysis planned. Good. Some remarks for group sequential design. As said, in contrast to fixed sample design, now the study protocol becomes a little bit more complex because the stopping boundaries, the number, and sample sizes of interim looks should be pre-specified in advance. And as said, the first group sequential designs were originally designed for equally spaced interim looks. That's the reason why nowadays we use mainly this alpha spending function with O'Brien-Fleming type of designs. On the slide before, I just introduced the concept using two-sided tests, but in clinical trials, we are mainly interested in demonstrating superiority of the new treatment against control. So, which actually means we are interested in one-sided statistical tests. Here I assume larger means better, and here you can see how the null hypothesis could be formulated. So that's the mean. The true mean is equal or smaller than the mean in the control versus the alternative. And the purpose of our clinical trial is to reject this null hypothesis. And the advantage of using one-sided test is that then you can easily use different stopping boundaries for both superiority and for futility. For example, for futility, you might stop when you observe a negative trend. And if you use a one-sided p-value, this would correspond to the situation where you would stop for futility when the p-value is larger than 0.5. Here is an illustration of the alpha spending function of Lanti-Mans. How does it work? It simply says pre-specify an alpha spending function and a maximum sample size. So the maximum sample size corresponds to an information fraction of 1. And the stopping boundaries is a continuous function of the accrual. And now the timing and number of interim analyses are quite flexible. This also allows for unequally spaced interim looks. And the increment in the alpha spending function between the two interim analyses is used to determine the critical boundaries and the correlation between the statistics. So by using this alpha spending function concept, you're really quite flexible because now you can do interim analysis whenever they become necessary. And how should you plan and compare group sequential design in the rare disease settings? Look at the power and the average sample sizes needed under the null hypothesis. So when there's no difference to placebo, for example, and for different alternatives, have a careful look at the stopping probabilities. On the one hand, stopping for futility, stopping for efficacy for each stage, and really think which number and timing of interim analysis is reasonable. Keep in mind, early interim analysis are not a very good strategy because there you have there's a data highly variable. You might be easily misguided by the data. So have a careful look at the minimum sample size, because the decision to stop, either for efficacy or futility, will be based mainly on the primary endpoint. And then you really think carefully about, is the first stage sample size large enough to check for consistency in as important variables, for example, and also calculate back which effect would be required for an early rejection. Should not only look at the p-value, transform it back into the effect size scale, because can such a large effect really be expected? If not, then there's no point of doing such an interim analysis if it's impossible to observe such a huge difference between the two groups concerned. Also have a careful look at the maximum total sample size, and there's also a discussion on binding, of using binding or non-binding futility boundaries. I'm just mentioning it here, I won't explain it, but the idea behind this is, if you use binding, stopping for futility boundaries, then you could gain in the critical boundaries for efficacy. So it's easier than to reject also for efficacy. However, you have to stick to this binding for futility boundaries. And especially the FDA suggests that it's better to use non-binding futility boundaries, which means you don't adjust the efficacy boundaries for the futility boundaries. Another issues which I will not discuss today is, if you're interested in the confirmatory testing of secondary endpoints, then you also have to foresee such group sequential boundaries for the secondary endpoints. Otherwise, the type 1 error rate will be inflated overall. Good. If you are interested to learn more on group sequential design, there are a lot of books out there. So here I just made a selection of 4. And for example, the book by Chris Jennison is a classic as far as group sequential methods are concerned. However, if more flexibility is needed, then you should use an adaptive design. So what's the difference between classical and adaptive trials? So for classical frequentist trial, all the details of the design and analysis must be prefixed in advance. So the population, which treatment are investigated, the doses, the analysis strategy, the sample size. And as already explained before, there's a clear lack of flexibility to react to information from inside or outside the trial. And that's the reason why people are interested in doing adaptation. So sometimes these trials are called adaptive designs or flexible designs, because this type of trial designs allow now for mid-trial design modification based on all internal and external information gathered at the interim analysis without compromising the type 1 error rate. So to control the type 1 error rate, even the design's modifications need not be specified in all details in advance. So today I will talk about confirmatory adaptive designs, where it's not necessary to fix all adaptation rules in all details in advance, and still you will control the type 1 error rate. So here some history of adaptive clinical trial designs. In 1989, Peter Bauer published his seminal paper on confirmatory adaptive design. The work was triggered by his work in the ethics committee and how amendments were handled or not handled at all in the statistical analysis. In the beginning, here in Europe, adaptive designs were considered as a kind of absurdity of academia and the German-speaking countries. And in the initial days, it was really hard to get funding for this type of design and also to get approval from regulators. But when more and more companies became interested in applying such adaptive designs, also regulators reacted by organizing workshops and issuing guidelines. For example, the EMA reflection paper was the first being published in 2007, and other agencies like the FDA followed. As there was a worldwide interest in adaptive design, now even on the ICH level, a guideline is developed, the ICH E20. And when people realized that regulatory agencies are open to discuss this type of designs, this really triggered a lot of research and also made it easier to acquire the necessary funding for adaptive designs. And if you are interested in the history of adaptive designs, look at the 24 papers, which is shown on the left bottom corner. And this paper was also discussed by regulators and by industry statisticians, and there you, I think, find quite a balanced discussion on the pros and cons of using adaptive designs. And if you want to know more on how adaptive designs proposals are received by regulators, you might have a look at one of the reviews presented here. On the left-hand side, you can see a paper on which scientific advices were actually given by the EMA Scientific Advice Working Party. And on the right-hand side, you can read what happened to these proposals in later marketing authorization procedures. And so you can also find some case studies with small populations in these papers. So which adaptations are of most interest in drug development? So this is again based on the review of EMA Scientific Advice Working Parties letters on adaptive design, and you can see one of the most attractive adaptations back then was the reassessment of sample size, but also treatment arm selection and population selection received some attention. However, there were also some concerns issued by the EMA based on this proposal. And you can see one main concern was that the proposals on using an adaptive design were not sufficiently justified. It is not clear how the potential bias by using an adaptive design is addressed later on in the analysis, and which was quite a surprise because there is really a broad range of methods available. There were really some severe concerns with respect to the control of the type 1 error rate, and also if too many interim analyses were proposed. So if we speak about an interim analysis where you might do some adaptation, then you should only go for one interim analysis. Otherwise, regulators might argue if you need a lot of adaptations during the conduct of the trial, then you haven't had sufficient information to even initiate the confirmatory clinical trial in the first place. So if we speak about adaptation in the confirmatory clinical trials, you should only go for one adaptive interim analysis. Let me show you some regulatory definition of adaptive design. So for example, if you go to the EMA guideline, it says a study design is called adaptive if statistical methodology allows the modification of a design element, sample size, randomization ratio, number of treatment arms at an interim analysis with full control of type 1 error. So you can see control of type 1 error is really essential if we speak about adaptive designs. The FDA guidance document for example, say a study that includes a prospectively planned opportunity for a modification of one or more specified aspects of the study design and hypothesis based on analysis of data, usually interim data from subjects in the study, and a clinical study design that allows for prospectively planned modification based on accumulating study data without undermining the trial's integrity and validity. And what is the minimum requirement of adaptive designs? So the EMA reflection paper says using an adaptive design implies that the statistical methods control the pre-specified adaptation error, that correct estimates and confidence intervals for the treatment effect are available, and that methods for the assessment of homogeneity of results from different stages are pre-planned. And the chief concerns with these designs are control of the study-wide type 1 error rate, minimization of the impact of any adaptation-associated statistical or operational bias on the estimates of treatment effects, and the interpretability of trial results. So just to summarize, do we have control the type 1 error? Do we have appropriate estimates for the treatment effect? And do we not endanger the interpretability of the trial results? And at the end of the day, when you think about such more complex clinical trial designs, you also have to write up the results. Then you might have to look at the adaptive design Consort extensions, which is an extension to the conventional Consort checklist when you would like to use the results coming from an adaptive designs. And in this paper, you can find a lot of case studies on adaptive designs for a broad range of adaptations. For example, if you look at table 1 in this paper, you can find case studies for sample size reassessment, also for case studies which use group sequential methodology as described before. Then there are also multi-armed clinical trials where you start with several experimental treatment arms. Response adaptive randomization is more controversially discussed. What are response adaptive methods? There you have a short-term endpoint, and based on the short-term endpoint, you change the allocation ratio time to time, so that overall you allocate more patients to the more promising treatment arms. Or if you go down, there are also examples, for example, for population enrichment designs. So just have a look at the checklist itself, but also on the case studies listed there. They're quite interesting. Now, I would like to give you the main statistical methodology on how to set up adaptive two-stage design. There are a lot of variations, but this is the main principle behind it. And I will illustrate it for an adaptive design where we do one interim analysis halfway through. So where I'm explaining an adaptive test procedure where a trial is performed in 2 stages. With stages, I mean I have 1 stage before the interim analysis and a 2nd stage with patients who have entered the trial after the interim analysis. In an interim analysis, the trial may be stopped for futility or efficacy, and if continued, then we possibly adapt the design, for example, the sample size, the test statistics, whatever. And the adaptation of the design of the 2nd stage, they really can depend on all unblinded interim data, including not only the primary endpoint itself, but also secondary endpoints, safety endpoints, and the adaptation rule needs not to be completely pre-planned. And how can we construct such a test that still controls type 1 error rate? There are 2 main principles: tests based on combination tests or tests based on the conditional error rate. And today I will focus on adaptive combination tests. If you're interested in the conditional error rate, just have a look at the 25 years paper, where you can also find some explanation on the conditional error rate. Good. So what are adaptive combination tests in a nutshell? So the main idea goes back to the papers of Peter Power from 1989 and 1994. So it essentially means you have to fix the design for the first stage. And in addition to a conventional trial, you also have to fix a combination function on how you combine the data coming from the first stage and from the second stage in case you decide to continue the trial based on the interim data. So for example, one well-known combination function is Fisher's combination test, where you would simply multiply the p-value coming from the first stage and second stage. So p times q. So p is the p-value from the first stage, q is the p-value from the second stage. Then we start the trial. When we get the data, we compute the p-value from the patients who were included in the first stage. And now if the p-value from the first stage data is smaller than a significance level alpha 1, we can already reject null hypothesis and stop for efficacy. If the p-value is larger than a predefined threshold alpha 0, then we stop for futility. So we have to retain the null hypothesis, and only if the p-value is between alpha 1 and alpha 0, then we decide to continue the trial. And now, based on all the data we have collected, we can fix the design for the second stage based on the data from the first stage. So only in the interim analysis, then we can fix, for example, the sample size for the second stage. Then we collect the data of the second stage, and that's the main difference to group sequential designs. In group sequential designs, we always pool the data we have accumulated so far, whereas in adaptive design, now you calculate a p-value for each stage, and then you combine the information via these p-values, and then you can see we don't throw away the data from the first stage patient, so patients who have been included in the trial before the interim analysis, because now the final test decision is being made on this combination function. And if this combination function C, which is calculated from the p-value P from the first stage and the p-value Q from the second stage, if this is small enough, smaller than a predefined threshold C, then we can reject the null hypothesis, and we have demonstrated that our drug is better than placebo. Good. Now you might wonder, despite all these adaptations, how are we able to control the type 1 error? It's quite simple. We choose this critical value C in such a way that this level alpha condition is kept for independent and uniformly distributed p-values P and Q. So this is just for the statistician here, because under the null hypothesis, we know that the p-values are uniformly distributed under the null hypothesis, and this characteristic is used when determining the critical boundaries. And as said before, a well-known combination function is Fisher's product test, where we simply multiply the p-value from the first stage P with the p-value from the second stage Q. So this idea was also used in meta-analysis, where you combine the information of separate studies into one conclusion. And here we have the information of separate stages. So these Fisher's product tests were initially quite popular, but over the year, there was a clear switch to another combination function, which is called weighted inverse normal method, which was developed by Gernot Wassmer in the paper of Lemacher-Wassmer 1999. And again, for the statisticians here. Here you take the p-value and transform them back to the Z scale, which has the advantage then we can use all the conventional group sequential boundaries, which is a nice feature. So we can pre-design our trial, for example, using an O'Brien-Fleming design, and by using this weighted inverse normal function, we can do also in addition adaptation, but the critical boundaries are exactly the same like they would look like in a group sequential conventional design. Good. So what is the clue of adaptive designs? The clue of adaptive designs is really we don't pool the data. We combine the information via stage-wise p-values. Because then the distribution of the combination function under the null hypothesis does not depend on the design modification. And hence, the adaptive test is really a valid alpha test, and we can convince regulators that we control the type 1 rate. And this simple principle can be now used for quite complex design. You can really expand it quite easily. You can use it for multiple looks, for multiple hypotheses, and so on. And the nice thing is really that the adaptation can depend on all data you have collected. That's really nice. And this surprises sometimes people. For a strict control of the type 1 rate, you even don't have to pre-specify how the stage 1 data to determine the stage 2 data from a statistical point of view, but still in the study protocol, you would fix also stage 2, so the most likely design you would like to have if you don't do any adaptations. Good. So let me give you one numerical example. So we have a small study in the rare disease setting, and we plan for 40 observations for 40 patients overall, and you can see the pair of hypotheses concerned. So we have the null hypothesis versus the alternative, and we go for one interim analysis. And for simplicity, just to illustrate the principle, I use now the Fisher's product test, and I use an alpha 1 of 0.01 without stopping for futility, which then gives me the critical boundary for the final analysis of 0.00326. And the interim analysis is conducted halfway through after 20 observations. And here you can see if the first stage data gives you a mean of 3.7 and a standard deviation of 10.9. When you use a t-test, you get a p-value of 0.0727. Now, let's compare the p-value with the efficacy boundary, but as the p-value is larger than 0.01, it means we cannot stop for efficacy. We continue, but now we can redesign our trial. For example, here, to be sure that we can reject at the end, we choose a sample size of 30 additional patients instead of the initially planned 20 patients for the second stage. So here you can simply do a sample size calculation, but you don't have to pre-specify the rule how you reassess the sample size. You could do it, for example, using conditional power arguments. Conditional power means how high is my power conditionally on the data observed so far, and then you can readjust the sample size so that the conditional power becomes 80, 90%, for example, again. But as said, by using this methodology, you don't have to tell people, is it the predictive power, is it the conditional power, or is it any other rule, ad hoc rule, which you would like to apply. Now, but you have to fix the second stage sample size after the interim analysis. So like here, we said 30 additional patients, then we collect the data, and based on this second stage data, we get a second stage p-value, which is as a p-value is 0.0376. And you can see for the final test decision, we use really all the data collected, and we combine the information via this combination function, And as this combination test statistic is smaller than the preset critical value of 0.00326, we can reject the null hypothesis, and we have demonstrated efficacy of our new drug. What about multiplicity in adaptive clinical trials? There are many reasons why multiplicity arises in clinical trials. So we could have multiple treatment groups, multiple endpoints, multiple subgroups. And in adaptive clinical trials, treatment groups, endpoints, and subgroups may be dropped or added in an adaptive interim analysis while controlling the family-wise type 1 error rate in the strong sense. What do we mean by control of the family-wise type 1 error rate in the strong set? That's the probability that any true null hypothesis rejected is bounded by alpha, regardless of which and how many null hypotheses are true. So that is the probability regulators are really concerned about. Now let me talk about adaptive seamless designs. So these are designs where I start with several experimental treatments, with experimental drugs, and at the interim analysis, I would like to select some more promising ones, because this type of adaptive seamless designs is quite attractive, especially for rare diseases, because then you can combine different phases of drug development into one trial and utilize the patients in a more efficient way. So for simplicity, I will just now show this principle, how you can use it to combine phase 2 and phase 3 objective into one trial design. So traditionally, you would expect that you have separate phase 2 and phase 3 trials. You can also call the phase 2 part as the learning part. Like you conduct a phase 2 trial where you might have several experimental arm and a common control arm. You can see on the left-hand side, you conduct this trial, then you stop, analyze the phase 2 data, and then based on the results coming from the phase 2 part, you plan your confirmatory clinical trials. So which treatments should be investigated in phase 3? What is this, how should the sample size look like? So you can see there's some white space between the end of the phase 2 part and the beginning of the phase 3 part. And for the very end, for demonstrating efficacy of the selected treatment, like C here, you are only allowed to use the data from patients who have been included in the phase 3 part. So you see, at the end of the day, you can only use this data, but you're not allowed to use this information for the final decision making, which is really a bit in the rare disease setting where you don't have huge sample sizes at all. So how do now adaptive seamless phase 2 and phase 3 trials look like? You can see now everything is done under one umbrella. So we conduct the phase 2 trial as internal part of a combined trial. And after the phase 2 part, we do an interim analysis. And in the interim analysis, still, we might do a sample size adaptation or and then selection of treatment arms. And the biggest difference is now at the very end, to demonstrate efficacy, we can now use all the data collected in the trial. So also the data from the patients included in phase 2 and also the data from patients collected in phase 3. Which means, what is the advantage? There might be a smaller time lag between phase 2 and phase 3, which might speed up drug development. It also allows us to use all the data for demonstrating efficacy, which helps us to save resources, costs, and time. And hopefully by using a joint protocol, it improves the quality of drug development at all. Just let me give you one example on such a trial. which is one of the proposals I have mentioned before, which is from the Elsässer paper on reviewing the scientific advice proposal on adaptive designs. So there was also one proposal on interim dose selection. So it was a proposal for a phase 2, phase 3 design for 2 pivotal placebo-controlled trials of a new chemical entity for the treatment of diabetic nephropathy. And the objective were to demonstrate superiority in a surrogate parameter, and the trial should start with 3 active arms, and based on the interim analysis, the more promising 2 experimental arms should be selected, and the dose selection should be done at all parameters available, so both on efficacy and safety. And the proposed step 1 error control was a Bonferroni adjustment, adjusting for the 2 selected comparisons only. So they started with 3 doses, 2 were selected, and the Bonferroni should be alpha divided by 2 because only 2 were selected. However, the statistical testing procedure was not endorsed, as it was not supposed to control the family-wise step 1 error rate for the 3 hypotheses initially considered, because if you select the more promising one, bias is already introduced, and instead adaptive combination test based on the closure principle and adaptive Dunning test procedures based on the conditional error rate have been suggested as alternatives. And the advantage of the proposed design with respect to power should be evaluated as it may be small. So you always also demonstrate that there is really a gain in power when using adaptive designs, because sometimes actually there might be a loss if the interim analysis is done too early and you are just misguided by the highly variable interim data. And Dimairo also had some concerns regarding the safety evaluation at interim. So which issues had to be addressed here? So again, control of step 1 error rate. Even if only one experimental arm is selected, a multiplicity correction is required, because you have to address for the fact that you might select the more promising one. Interim selection of treatments might introduce bias, and if the treatment with the largest interim effect is chosen, the effect estimates will be biased, and interim data might be highly variable and lead to selection of the wrong treatment arm. And clearly, it also requires unblinding of the data, which might compromise the whole trial itself. And for the control of step 1 error rate, several procedures have been proposed. Methods based on completely predefined adaptation rules, or using such adaptive combination test, what we have discussed before, in combination with the closure principle. I will explain this closure principle in connection with adaptive combination tests here. So again, the selection of treatments may depend on all data collected, and we would like also to adapt the sample sizes. And in principle, pre-specification of the adaptation rule is not required to control the multi-step 1 error rate. However, the type of adaptation and anticipated adaptation should be pre-specified in the protocol, and the number of adaptation should be limited. So I will illustrate this principle again for a multi-arm clinical trial design with 1 interim analysis, and we start with 2 doses and a control group. So in total, we have 3 parallel groups, and we're interested in testing the one-sided hypothesis. So the first one is that treatment 1 versus control, and the second one is treatment 2 versus control. And after stage 1, we could decide either to select both treatments or just one. However, we don't know the exact selection rule, because we will look at both efficacy and safety data, and also the choice of the sample size for the second stage depends on how many treatments we actually select, and also on the observed efficacy. And for the control of the type 1 error rate, it's not sufficient just to use this adaptive combination test for each single hypothesis separately. We also have to look at the intersection of these 2 null hypotheses concerned. And now we can also use an intersection, also for this intersection test, a combination test. And how does it look like? You can see again, we use this combination function, CBQ for the intersection. And for the first stage, we use a multiplicity adjusted p-value because we're testing treatment 1 and treatment 2 against control. And for stage 2, so for Q, it depends. If we select only one treatment, then it's simply the second stage p-value of the selected treatment. And if we select 2 treatments, then again we have to use a multiplicity adjusted p-value. Like here you can see the equation for the CDAC test. And in any case, we reject this intersection hypothesis if the combination test statistic becomes smaller than the predefined level C. So what do I mean by the closed testing principle? So the closed testing principle means you not only have to perform a statistical test at level alpha for each elementary null hypothesis. So on the left-hand side here, this is for treatment 1, on the right-hand side for treatment 2. And in addition, we also test the intersection as outlined on the previous slide. And the intersection and the closed test principle simply says to reject an elementary null hypothesis, you can reject it if the test for the elementary rejects and all intersection hypotheses where this specific elementary hypothesis is part of. So it means if the test for the intersection can be rejected, none of the elementary null hypothesis will be rejected. But the advantage is if the test for the intersection can be rejected, then we can test all elementary null hypotheses full level alpha, and no additional price for multiplicity has to be paid. And here you can simply see the statistical test here then simply becomes a combination test. Good. In conventional clinical trial, the power is a quite nice and clear concept. However, how to define power if multiple hypotheses are tested in an adaptive trial? Clearly think of what are you actually interested in, because this can have a huge impact on the sample size needed. For example, you can be interested in the probability to reject all hypotheses, the probability to reject all selected hypotheses. You could be interested in the average power for the selected hypothesis. The power could be defined as the probability for a particular treatment to be selected and the corresponding hypothesis to be rejected, or the probability to select and reject any hypothesis. And clearly, if you go for the red one, then the smallest sample size is needed. So it really has an impact on which power definition you use. So let me give you an example for an adaptive seamless design where you start with 2 treatment arms and you select the more promising one. And the green line would be a classical done test where you always select post-treatment, but let's say this is not possible because it's interim analysis. For ethical reason, you can only select the more promising one. If you would still perform a classical done test, you see that you have quite a huge drop in power. By using the adaptive combination test principle, you can see we have already a higher power. This is a straight black line. However, if you use this adaptive methodology and reallocate the sample size of the dropped treatment to the selected one, then actually we have a huge power gain, which is expressed by the dash dotted line. And this is a power to reject at least one of true, to receive at least one true rejection. So just by combining the very basic adaptations of selecting the more promising one and relocating the sample size, you can see we can gain a huge gain in power, which might be a better use of resources. Good. As you might correctly say now, yeah, but in clinical trials, we are not only interested in testing primary, but also in interesting testing secondary endpoints, you can use this principle and become more and more complex, like we have adaptive graph-based methods where you can test both primary and secondary endpoints, and these graphs then defines your adaptive tests in the background, and you don't have to care about how the weighting is actually done. And the same principle can also be applied for adaptive enrichment designs. So what are adaptive enrichment designs? Say you start with a broader study population, and based on the interim results, you decide either to select a more promising subgroup and just recruit patients from the more promising subgroup for the second stage, or you might decide to continue with the full study population, and then in addition you might decide either to test efficacy just in the full population or both in the full population and in the subgroup as well. So you can be quite creative in applying this principle. Here are some references for adaptive designs. Have a look, for example, at the book by Gernot Wassmer and Werner Branner, or again, have a look at the 25 years papers of confirmatory adaptive design, what I had already mentioned. Now, in the end, I will come to even more complex clinical trial designs under the term master protocols. So let us first fix the terminology on master protocols, where we have basket, umbrella, and platform trials. So let's have a look on which definition Jen Woodcock and Lisa Lavange used in the fundamental paper on master protocols. So basket trials, you have one investigational treatment, is evaluated in the context of multiple diseases or disease subtypes with a common therapeutic target. Then we have umbrella trial, multiple investigational treatments or combination are evaluated in the context of a single disease, possibly with several sub-studies for different disease subtypes. And then we have platform trials. It's an umbrella trials where drugs or combinations may enter or leave the trial throughout the conduct of the trial. And now we will focus a little bit on these platform trials. If you're interested on learning more on master protocol, have at the review showing, have a look at the review from Meyer et al., which gives a good overview on both on the methodology and also on which trials have been already conducted. And now let me go back to the figure we have started with in the very beginning. As said, traditionally, the type 1 rate is controlled at study level. There's no data sharing across studies, which is a little bit in rare disease. However, the sample size and power calculations are quite simple, but we don't share information across studies and indication. And there really comes the gains for rare diseases. And why is there a wish for something different? Because it's really an inefficient use of resources. Standalone RCTs needs their own control group. Each time you have to develop a new protocol, a new SRP. You have to get ethical approval, regulatory approval for each trial itself. You have to look for appropriate trial sites. So it's really time consuming to set up each trial on its own. And therefore, there's really interest in such collaborative platform trials. What are the design characteristics of platform trials? So you start as a multi-armed clinical trials, then you have several interim analyses, and in the interim analysis you allow for adaptations, like you might change the allocation ratio based on response adaptive methods, where you increase the allocation probability for the more promising treatments. The advantage is really that you can share the control arm, and also new treatment arm can come in later on, as shown here on the slide. However, as these platform trials can run for a very, very long time, you might be into the situation that you start with a control group, but out of a sudden, when a drug is approved in an indication, so let's say the placebo control group becomes unethical, so there might be even a switch of control group. So even though there might be some efficiency gain, it comes also with some hurdles. So what are the advantage of platform trials. More patients are eligible for a trial due to multiple treatments and substudies with possibly different inclusion criteria. By using a joint trial infrastructure, you might save time and money. From statistically, you test multiple hypotheses in the same trial, which is also a challenge in itself. And the real gain comes by sharing of control data and by using adaptive decision rules, which hopefully leads to needing fewer patients, and it also allows for a direct comparison between treatment arms. However, what do you think? Are there some multiplicity issues? Here you can see the sources of structural multiplicity in platform trials, because now you are testing multiple drugs. You might even have multiple treatments per sub-study. You might have multiple adaptive interim analysis. You allow for data sharing across these sub-studies. But the main question is, do you have to adjust for multiplicity as you would like to test drug A versus control, drug B versus control, drug C versus control? And in the past, regulators would have argued, okay, if it's one trial, you have to adjust for multiplicity. And then it wouldn't be very attractive, especially in the rare disease setting, to go into such a trial, because instead of, let's say, using 5%, if you plan that overall up to 10 treatments might come in, you have to use a multiplicity correction, and instead of 0.05, you have to use 0.005, a significant level, then you have no chance to find anything. So is there a need to adjust for multiplicity in platform trials? And the answer is no need to adjust when the hypotheses are inferentially independent, and hypotheses are inferentially independent if the truth or falsehood of one hypothesis is unrelated to the truth and falsehood of the other hypothesis, which means when no extrapolation from the rejection of one hypothesis to the other is possible. And the main argument is really, if we did separate trials, we would also not adjust for multiplicity. So why should we do it in the context of a platform trial? And here on the right-hand side, you can see what is meant by inferentially independent. Independent means when we have different drugs with different mechanism of action or different drugs with similar mechanism of action. However, if you test different doses of one drug, then still an adjustment might be reasonable. So what's a pragmatic strategy for statistical inference? Each sub-study in the platform trial is considered as independent, and for each you can use the full level alpha as you would do in a standalone study. However, you would still adjust if you're interested in testing several endpoints. You would use the adaptive methodology if you do adaptive interim analysis, but you don't have to adjust that several sub-studies are ongoing in parallel, and it doesn't matter that you share the same control data. It doesn't matter. So the concept of study-wise step one rate is not directly applicable to platform trial, especially if they are perpetual in nature. As said, it really depends how independent the inference is. Still, you could argue that there's not a complete consensus on what to consider independent, but it seems that no adjustment is needed, that several sub-studies are conducted within the same master protocol. And let me now elaborate a little bit on sharing the control data, because the question is really, can we use all control data which is already available? Think of a platform trial where you start with treatment A versus control. This treatment starts, and later on a new treatment enters as well. And if a platform trial runs over a long time with multiple treatments entering and leaving the platform trial, incorporating the non-concurrent controls can substantially improve the efficiency. So what do I mean by non-concurrent controls? It means we have on the one hand side the concurrent controls for treatment B, which is the control data which were collected in parallel when treatment B was already in the trial. However, we have also the non-concurrent control for treatment B, which are the control data which have been collected before treatment B enters the trial. So non-concurrent control data exactly the same as using historical controls in randomized control clinical trials? Nope, because you have a higher data quality. Because the data are collected within a framework which has many features standardized, the same infrastructure, the same assessment of endpoints, the same monitoring, and all changes are well documented. And non-concurrent controls can also be randomized and blinded, but, and that's the tricky part, at a different calendar time, such that randomization doesn't ensure control on the distribution of prognostic factors between the non-concurrent control and the experimental arm. And this lack of true randomization can really induce time trends. So there could be time trends due to external and internal factors. External, for example, the patient population can change, the standard of care can change, or there could also be internal factors like over time, the recruiting centers can change, or even the assessment of endpoints could change, which could, due to new diagnostic devices available, which could also mean that we see some time trends. So can we use all the data? The problem is naively pooling control data can lead to an error. So here you can see the blue line is where we just take all the data we have for treatment B. And you can see then we can have a huge power gain compared to the green line where we would only utilize concurrent control data. But when you look at the right-hand side, depending on the time trend, which is shown on the x-axis, it could mean that actually the type 1 error rate is inflated, depending on the time trend. And again, then regulators would become nervous. However, the good news is by regression models, the red line, you can adjust for time trends. There are both frequentist and Bayesian methods out to adjust for possible time trends, but you have to be aware of what are the assumptions behind the regression models. Because again, if the assumptions are violated, this can lead that the type 1 error rate is actually inflated. So carefully discuss whether there could be time trends, and based on which time trends you expect, you can then refine your regression model. If you're interested more in this topic, have a look at the paper by Marta Bofill, which was just recently published. So a short summary on using non-concurrent controls. The inclusion of non-concurrent controls is a question of variance bias trade-off. Methods to address potential biases are available. However, they rely on specific assumptions, and especially in the rare disease setting, it might be reasonable to go for such a modeling approach to increase statistical efficiency. Good. Let me give you an example for such a platform trial in the rare disease setting. It's a proposal for a platform trial for neurofibromatosis coming out of EU Pearl. And you can see you have your master protocol where patients might first be included in an observational cohort. And depending whether treatment options are available or not, patients might be randomized to a sequence of available treatments. And if a patient meet this criteria, he is then treated with the randomized drug until the criteria for treatment discontinuation is reached. And then again, it's proved if the patients can be reused and re-randomized, or if it is the case that the patient is not longer or maybe based on the inclusion criteria, the patient is not longer eligible for the master protocol. And by reusing the patient again and again, you might increase the statistical efficiency, and the proposal has been set up in such a way that you can even start with just one treatment arm, and depending when other treatment option becomes available, you expand this platform trials. So have a look at this paper. If you want to learn more on platform trials, have a look at a summary paper coming out of EU Pearl, which was an European project learning more on platform trials. You can see the link here. And I would especially highlight that there are also templates available on how to write a master protocol and how to write the protocols for the sub-studies, which are called intervention-specific appendixes. And if you go to the online supplement of the paper by Miesenbrink et al., then you can find really Word documents which you can use to write your own study protocols. Good. If you're interested in software for adaptive designs and master protocol, simply have a look at this systematic review shown on the left-hand side. So there's a lot of adaptive software out, both commercial and non-commercial software. Commercial, for example, AdPlan or Facts, and non-commercial software in R like RBACT or GS Design, for example. And for platform trials, you might have a look on the papers provided on the right-hand side. Good. Let me give you some conclusions. So adaptive clinical trials might make a better use of the resources available compared to traditional parallel group designs, as they allow for mid-learning and adaptations by strictly controlling the multiple step failure rate. Platform trials and master protocol are pose a challenge and an opportunity, but due to sharing control data, sharing infrastructure, there might be a more efficient way forward. And when you think about implementing adaptive design, have a look if you can answer this question, because this you should be able to address during planning and assessment. Is there a good rationale? Have alternative, more standard trial designs be considered? Does the proposal fit well in the context of development program and the data that will be available for the marketing authorization application? Can the proposal be implemented without important damage to trial integrity? Is the type 1 error rate control? Has the potential bias of treatment effects estimates been evaluated? And is the proposal practical and feasible? And adaptive design seem well accepted if properly planned and implemented. A range of increasingly complex adaptive designs are proposed, the majority in rare diseases. Surprisingly, still a lack of methodological knowledge on how to achieve the type 1 error control, and also how to assess the efficiency of the design by using clinical trial simulations. And a clear question is, who should decide on adaptation at interim? The data monitoring committee, the sponsor, and group sequential designs presented in the beginning of the presentation have been developed in the 70s and are now well established. And the question is, when are adaptive designs commonly accepted? Here, some references, and all three papers have a long list of references where you can find more on adaptive designs, group sequential designs, and master protocols. And I should also acknowledge that the slides presented on platform trials go back to the EU Pearl project I had mentioned before. So many thanks for your patience. And if you have any more questions, just drop me an email. Thanks a lot.